Good morning, everyone. I am back, and this time I wanted to give an update on a article I did before about a month ago. I'll leave a card or a link to that video in the comment section or the description box, so just look out for that. But it was on I-Motif DNA structures, and this comes from Cancer Discovery. It's well renowned as a source for research on cancer, obviously, but also on discoveries that are not related to cancer that can be twisted and turned into something that's used to either treat cancer, but in my interests would be more on curing the disease itself. As we know, cancer and most diseases, viruses as well, cannot survive in an alkaline environment. It's difficult for them to survive in an alkaline environment and reproduce in that environment. But when you look at the DNA structure, you have to understand that certain DNA structures can make you susceptible to those viruses or bacteria that are coming in. They can also, those infections can also stimulate regulatory changes in the DNA itself. So in this article, they discussed that I-Motif DNA structures may regulate promoters and telomeres. So promoters are pieces of DNA prior to the open reading frame of a gene that help to reproduce that gene or help that gene to transcribe RNA, particularly mRNA or messenger RNA. Messenger RNA can stop there and just live forever as messenger RNA, or it can be used to translate itself into proteins. Proteins, of course, being very important for most biological processes. Now, telomeres are pieces of DNA found at the tips of chromosomes, and those pieces of DNA are involved with aging and also involved with cellular replication. So all of this is all mixed together. Now, if you go to Keith Newsom, I think he has discussed DNA, if I'm not mistaken, way back in the day. I may be mistaken about it being Keith, but I believe he has discussed this type of, just briefly, with DNA. He discussed blood types. I do remember that. And blood types also is related to your DNA. And they're finding that more and more relevant to the diseases that we're faced with on a daily basis. So let's get into this article. What you'll notice here, sorry, there's an ambulance going by. Okay. What you'll notice here is that there's no article. Where's the freaking article? So if you come to this page, you won't find an article. If you click PDF, it won't let you download. It'll say log in using your username and password. Or you can purchase, which I'm not going to do. So yesterday there was an issue with me finding more information concerning that particular article. What I'm going to show you is how you can find their data without purchasing their articles and without even worrying about downloading their PDFs. So I'm going to go to another page here, and let's see. I'm just going to actually go back. I'm going to copy the title, copy that, and I'm going to go to Google. I'm going to paste in their title. And what I'm going to do is, because they have it listed all down here, but you're not going to find, it's hard to find their whole article. Let me see. Maybe I can find it here at NCBI. I'm going to try to see. It says find the full text at the journal site. That takes me right back to where I was. They're not going to help me. Okay, no problem. Click on images. You can find their images here. And all you have to do, let's see, come down some more. So we can find, let's see, their website. It was published on Cancer Discovery. Okay, so we just remember Cancer Discovery. It's probably going to be CD. It was AACR. AACR is the American Association for Cancer Research. And I think anybody can become a member of that if you wish to. But I'm gonna just click Wikipedia because I clicked this earlier. So this gives a structure of the G4 DNA structure and motif. So it's rich in hydrogen, nitrogen, oxygen, and of course the lines represent the carbons, the R factors that you have sticking off the edges as well. That's in figure A. So this is a structure of a G quartet. This is a tetrahedron structure, ring structure. So it is a closed system. In B, they have a schematic of a intramolecular G4 DNA structure consisting of 3 G quartets. So they stack on top of each other with 3 loops that represent a pyramid structure. If you can see, you have loop 1, loop 2, and then loop 3, 1, 2, 3, like that. Or 1, 2, 3, going that way. Upside down pyramid that represents the loops. So this is all geometrical. Now, if you think about the ancient Kemetic scientists and the Dogon, they already knew about these principles. 5 prime, 3 prime ends, the quartet tetrahedron G structure that is linked on top of each other. And what they've done is taken A and stacked it in a picture form, and that's how they get B. So you have an open picture, it's laid flat in A, and they've allowed it to be formed into a box structure because that's how it's formed. In C, they stretched it out and showed the loops with the G4 G tracks that are shown there. If you come to Wikipedia, let's see, I'm going to go to this link, visit the link. It is in Wikipedia, but they have, let's go down. They have references here, tons of references that span from 2011, 2008, all the way back to 2005, 2002. These are all references. If you notice, you don't see a lot of our names listed here, so this makes it more important for us to be in science. You see a few people with, I guess, quote-unquote American type names, but most of them are Asian, East Indian, and Caucasoid. So this is just talking about the background. What does it mean? What is a G quadruplex? A G quadruplex is based on guanine. Guanine being a nucleic acid that's important for the structure of DNA. Phosphate is also important for the structure of DNA. It keeps it in a negative composition and therefore giving it a negative frequency. So anything that binds to DNA, such as proteins or maybe fat, anything that's liposomal that could bind with DNA, or anything that would interact or try to interact with DNA, would either not have a charge or have a positive charge, usually a positive charge, such as protein coupled receptors, protein that would bind DNA in some kind of way, or RNA. So they come down and they say here, where is it? At the end, it says G quadruplex structures can be computationally predicted from DNA or RNA sequence motifs. but their actual structures can be quite varied within and between the motifs, which can number over 100,000 per genome. That's a lot. Okay, they say their activities in basic genetic processes are an active area of research in telomere, gene regulation, and functional genomics research. And so before I go further, I also wanted to give you a visual representation of telomeres. Let's do images. Oh, let's see. Let me do all first, just to give you a brief definition. A telomere is a compound structure at the end of a chromosome. That's the fast definition of it. So they have an image here. So it's very important, and they show you here. Telomeres are the end caps that protect the chromosome. They keep it closed. If you did not have telomeres, the chromosomes would unravel and you would not be born, or you would die. So they're needed. As the cells divide over time, telomeres shorten, and eventually the cell division will stop. So it does have an effect on aging. This is why telomeres are studied during Alzheimer's research. They're also being studied now for diabetes research, and of course they're being studied for cancer research, because if you can control telomeres, you can control how the cancer divides. You can actually stop cancer division and put cancer in a senescent state. So the tumor cells don't go away, but they stop growing, they stop dividing, because that's the number one problem for cancer cells. As I said earlier, you can have cancer cells that don't grow very well in an alkaline environment, but they can go through a hypoxic and protective stage where they still grow, but they're not growing as fast. They can control their DNA damage due to alkalinity. They can control their DNA damage by continuing to grow in a very dangerous environment, because the environment that's natural is dangerous to cancer cells or virus cells, right? So this shows one cell dividing and becoming 2 cells, 4, and then going on, each of those cells dividing into more. So 2, 4, 6, 8 cells, and then to 16, 32, and 64, so forth. So as these cells divide, the telomeres eventually get shorter and they stop division. And there's tons of pictures here to show that. They actually say exercise is proven to slow down the telomere shortening. And they've done a lot of exercise studies when it relates to, of course, depression, Alzheimer's, dementia, cancer, diabetes. So this is just important to note what is being studied. Now they say here you see the picture again with the G it's really a quadruplex that they have. They have the R factors, and now here you have a crystal structure. So this is protein crystallography or structural crystallography, where they have put in the sequences of this G quadruplex or G tetrad, and they've allowed it to go into a computer system, and they're modeling what is happening. And what they can do is add in different stimuli to see how the structure changes over time. They can predict how cancer cells would react with a G quadruplex affecting certain genes. So it says here in the description, it's a 3D structure of the intramolecular human telomeric G quadruplex in potassium solution. The backbone is represented by a tube, and that's that gray and green tube. The center of the structure contains 3 layers of G tetrads. The hydrogen bonds in these layers are represented by blue dashed lines. So hydrogen bonds, you have hydrogen bonds, covalent bonds, van der Waals interactions. The hydrogen bonds are pretty strong when it comes to a G quadruplex. However, these bonds can be broken based on acids or radiation or different stimuli that are designed to be destructive. And I just thought it was interesting to give an update on what they've been studying in terms of this I-Motif. So if we come down here, they have other tabs you can click on: the structure and functional role in the genome. And they give those, they give the Hoogsteen base pair bonding, which is a theory of how the base pairs adenine, guanine, cytosine, and thymine, how they're binding to one another. And they come down here. They do talk about the repair and DNA damage, as I mentioned earlier. It says, because repair enzymes would naturally recognize ends of linear chromosomes as damaged DNA and would process them as such to harmful effect for the cell, clear signaling and tight regulation is needed at the ends of linear chromosomes. And then they say telomeres function to provide this signaling. We know that telomeres are rich in guanine and with a propensity to form G quadruplexes. They're located at the terminal ends of chromosomes and help maintain genome integrity by protecting these vulnerable terminal ends from instability. So you can get instability, as I said earlier, and I am also getting help from Ajali as well on this topic. I'm really glad that I'm able to watch other people's shares and videos and learn more about what I study anyway. So they have tons of articles here. I really appreciate other people's work, and Ajali, Keith Newsom, and many others. I apologize. Lisa Cabrera as well, True Royal Family. I apologize if I'm missing anyone when it comes to science and technology. But I just typed in chromosomal instability from radiation. And you have tons and tons of articles here from 1998. Radiation-induced chromosomal instability in human T cells. Spontaneous and radiation-induced chromosomal instability. Numerical chromosomal instability mediates susceptibility to radiation treatment. So that means the more unstable your chromosomes are, the more susceptible you are to radiation treatment. And that's for patients receiving radiation. So as you can see, it just goes on. And I really encourage people to just search for things. You know, it may seem crazy, the topics that you want to search. Trust me, there's nothing crazy when it comes to science. Everything can be searched. Trust me, there's an article out there on it. You know, you can make up letters if you want and see if it works for you. I'm going to do that now just to show you. Oops. I keep getting this little ad here. I'm going to just make up letters and see if a gene pops up. FDG gene, just to see. And here you go. I just made it up. FDG, 18F-FDG kinetics and gene expression in giant cell tumors. I don't know what that means, but I would be very inclined to look it up. Now I see it's about immune cell microenvironment in head and neck cancer. FDG. Didn't know that before just now. Okay, let's just make something up. I'm just typing in random letters. I'm trying to make sure there's no vowels. So the LWYFW. Just kind of made it up. Now this is getting some other crazy stuff with, all right, malware. That's something different. I don't want that. Let me type in something else that's not so weird. PISDGR. Okay, PISGR. Okay, PISGR. Okay. So I just typed in something randomly. Okay, I'm getting things that are in other languages, but you can understand where I'm coming from. So you can type in almost anything and get something back. let's see, DQU gene. Let's see what I get. So I get microRNAs. Maybe there's something that matches DQU. Okay, there's a HLA-DQ region. So that's very interesting. There's celiac genes that relate with the celiac disease. They have DQ polymorphisms. That may be interesting to you. Evolutionary or evolution and genetics of populations. That may be interesting as well. Probably dealing with some, it has some equations. So, like I said earlier, take time to just search for different things concerning science. It's important for us to know what's going on in the world around us. I hope you enjoyed my video. I hope it wasn't too long. I'm not here for, self-aggrandizement or anything. I'm here to help other people learn more about science, and hopefully my main goal is for us to learn together and to be more aware of what's happening in every aspect of life. And I really encourage you to, step out there and look for new things. I mean, you'll be surprised at what you find, and the interesting things that are being studied right now. So that's all for me. I don't want to keep going on. You all have a great day, and I'll see you in the next video. Peace.